It's a huge honour to be made a Fellow of ISFAL. This is a really important society to the field of fatty acids and lipids, and to me personally. And to be recognised in this way by the society is very humbling. I think it gives some recognition of the work that I've done and my group has done in omega-3 fatty acids in particular, but also in other fatty acids over a long period of time. And it's really great that the society recognises our work in this way. I first became interested in omega-3 fatty acids in 1987, when as a new postdoc I went to the Department of Biochemistry at the University of Oxford, and I was assigned a project to look at the metabolism of fatty acids in macrophages. At that stage I knew very little about fatty acids, so I had to do a lot of reading on the subject, and I came across really interesting papers on omega-3 fatty acids. And I thought I would include omega-3 fatty acids in my studies of fatty acid metabolism and the macrophage. It turned out this led to a path of research which continues to this day, because in those early experiments of omega-3s, I found that they were able to influence basic functions of macrophages, like phagocytosis, adhesion to surfaces, and so on, and that the properties of omega-3 fatty acids were very different from the properties of other fatty acids, particularly saturated fatty acids, but also omega-6 fatty acids. So this led to a whole research program over the years comparing omega-3 fatty acids with other types of fatty acids. My work on macrophages led on to work on T cells, and I was able to identify differential effects of omega-3s on T cells, particularly on cytokine production by T cells and T cell proliferation. And I linked those effects on T cells to differences in membrane incorporation of fatty acids and membrane fluidity. These early in vitro studies ultimately led to animal experiments and then on to human experiments, and as I say, into a lifetime of research interest in these interesting omega-3 fatty acids. I think many of our most important findings on omega-3 fatty acids, particularly with regard to inflammation and immunity, come from the early in vitro studies. So I was able to describe the relationship between omega-3 fatty acid incorporation into macrophages and the ability of macrophages to phagocytose different particles, and went on to look at omega-3 fatty acids in T cells, linking the ability of omega-3s to influence T cell proliferation to changes in membrane fluidity of those cells. I was also the first to show that omega-3 fatty acids directly affect cytokine production by T cells. These early functional studies led to more mechanistic investigations, and one of my PhD students was able to demonstrate that omega-3 fatty acids influence really early signaling events in T cells, including the phosphorylation of the important signaling protein phospholipase C gamma 1. And in the paper describing this effect on the signaling process, we predated the later discovery of lipid rafts with our explanation for how omega-3 fatty acids might affect signaling in T cells. In addition, we made very important discoveries around the influence of omega-3 fatty acids on adhesive interactions. Again, studying macrophages, this time taken from animals fed different diets, including a fish oil diet, providing a lot of omega-3 fatty acids, and how well those macrophages would bind to endothelial cells, a very important adhesive process initiating inflammatory responses and also the development of atherosclerosis. So we were able to identify effects of omega-3 fatty acids on key adhesion proteins and on the adhesive process. Finally, I think in human studies, some of our work looking at the dose-dependent and time-dependent incorporation of EPA and DHA into different pools in the body, including blood lipids, blood cells, and also tissues, has been really important in terms of defining dose-response relationships to omega-3s and how they might affect human health outcomes. I've been lucky enough to be involved in some really interesting and novel projects in both experimental animals and in humans. For example, we did a project once in so-called nude mice. These are mice that lack a thymus and therefore can't mount a very good T-cell mediated response. Those mice are able to grow human tumours because the immune system that exists in the mice won't reject the tumours. So we did a very interesting study looking at the effect of feeding those mice omega-3 fatty acids on the growth of human colon tumours implanted in athymic mice. And we identified that compared to feeding vegetable oil, feeding fish oil to those animals resulted in smaller tumour growth, which I think is very interesting, indicating that omega-3s could have a role in preventing tumour growth. In clinical trials, perhaps the most interesting study that I've been involved in was looking at whether omega-3 fatty acids can get into advanced atherosclerotic plaques, which we demonstrated they could, and that once they're in those plaques, they exert anti-inflammatory effects that serve to stabilise the plaques and make them less likely to rupture. We think this could explain why omega-3 fatty acids have an effect on preventing coronary heart disease and other vascular disease. Also in humans, I was involved in a very interesting trial of intravenous infusion of omega-3 fatty acids into critically ill patients, where again we showed a decrease in inflammation, which influences the clinical outcome in those patients, and this was linked to better lung function and to a reduced length of hospital stay. More recently, I've been involved in interesting studies of omega-3 fatty acids in patients with non-alcoholic fatty liver disease, a very common condition that is increased with increasing obesity. And we demonstrated that omega-3 fatty acids could target liver metabolism of fat and also liver inflammation. And this can reduce the amount of fat in the liver of those patients, indicating a new indication for treatment by omega-3 fatty acids. Of course, research in omega-3s has been going on for decades now, and we know so much now about the effects of omega-3 fatty acids and their mechanisms of action. But of course, there are many new discoveries that await us in the future. I think in recent years, the important discovery of the role of omega-3s in resolution of inflammation has really revolutionized research in this area, and perhaps explains the effects of omega-3s in many, many different areas that have already been described. So I think going forward there will be really interesting work, perhaps discriminating between the effects and the mechanisms of action of EPA and DHA, and investigating how DPA can also have biological actions. I think because these fatty acids come from seafood, creating some issues around sustainability, that there'll be further research into more sustainable plant alternatives for bioactive omega-3 fatty acids. These could be other omega-3 fatty acids from plants, or they could be non-omega-3 fatty acids that have high biological availability. I think one of the other interesting things is that omega-3 fatty acids don't always have the same effect in a particular group of individuals. And understanding the reasons behind the variability in response to omega-3s, some of which could be genetic, but others could be lifestyle factors, other dietary factors, to try to take greater advantage of the effects of omega-3s and personalize their application for both prevention and treatment will also be a very important area for the future. And I think finally for the future we're going to need much bigger and better human trials, because many of the human trials that have been conducted to date haven't really provided the convincing evidence that preclinical research says they should. And maybe one of the issues is those trials just haven't been big enough, haven't been well enough conducted. So in the future, I think this will be a key area for research in our field.